Today we will talk about the morphological variants of necrosis. We have mentioned in the theoretical part last lecture that there are five types of necrosis, four morphological types of necrosis. These morphological types of necrosis are characterized by different appearances. Whenever the tissue dies, whenever the cells die within the tissue, they will take a special type of appearance called the morphology. يعني احنا نقول appearance نقول morphology بعد ماكو شيء اسمه appearance نقول morphology. This appearance of this morphology is dependent on the mechanism of necrosis that takes place. And the essential mechanism of necrosis depends on two mechanisms. The first mechanism is coagulation of proteins that happens during the course of cell injury, during the course of cell death. And the second mechanism is digestion of cell components or cell contents by lysosomal enzymes, واللي نصح له بين قوسين autolysis. All cells, whenever they die, they will start to digest its contents by release of its lysosomal enzymes. And then these two processes, they compete with each other. يعني اكو فد عملية تنافس بين وحدة والثانية. إذا صارت وحدة أسرع من الثانية راح تعطينا special character, special morphological feature يختلف عن الثاني. يعني إذا صار coagulation سريع أسرع من الlysosomal enzyme activation و lysosomal digestion, هنا راح ينطينا special morphological feature يختلف عن إذا صار العكس تماما. The first type of necrosis is coagulative necrosis. Coagulative necrosis, من نقول coagulative نقصد أنه there is coagulation of proteins, واللي هو يعني in another term refers to denaturation of proteins. Denaturation of proteins. Whenever coagulation of proteins happens rapidly, more rapidly than the lysosomal activation, this will lead to coagulative necrosis and is classically seen in myocardial infarction. يعني the classical example of coagulative necrosis is myocardial infarction. In this type of injury, in this type of necrosis, the cells are preserved. The architectural features of the cell are preserved. So they are mummified, أصير على شكل يعني خلينا نقول صاير بيها تحنيط متحنطة, mummified, and the cell outline is preserved. The cell cytoplasm is preserved مثل ما هو. There are special features that refer that these cells, although they are preserved in morphology, but we have special features that refer that these cells are dead. Among these features include مثل ما ذكرنا بالمحاضرة السابقة أنهم the first cytological feature to appear during the course of cell injury is cell swelling due to influx of water from outside of the cell into the cell. Cellular swelling. The second feature is cytoplasmic eosinophilia. Cytoplasmic eosinophilia, which is due to three causes. The first cause is accumulation of lactic acid leading to cytoplasmic acidosis. The second cause is detachment of rough endoplasmic reticulum from the detachment of ribosomes from the endoplasmic reticulum, leading to deepening of eosinophilia, يعني صار لون الخلايا أحمر. And the third cause is accumulation of inorganic phosphate due to degradation of ATP to ADP, leading to accumulation of inorganic phosphate. والinorganic phosphate بعدين بالأخير راح يتفاعل ويا المي ينطيني phosphoric acid, and this will lead to deepening of eosinophilia. As you can see here in this photomicrograph, there are myocardial cells. They are deeply eosinophilic, لونها أحمر طوخ. هاي كلها عبارة عن myocardial cells. All these are myocardial cells. They are deeply eosinophilic. They have preserved cellular outer contour, يعني شكل الخلية محتفظة به. هذه همين نفس الشيء. كلها they have preserved cellular outer contour. The cell size, the cell cytoplasmic contents، يعني ذن كلهن محتفظات بهم. بس شنو اللي نقدر يعني نميز به عن طريق مجموعة من التغييرات؟ شلون نقدر نميز أنه هذه الخلية ميتة؟ اللي هي بهاي الطريقة: cytoplasmic eosinophilia, cellular swelling, and with the progression of cell necrosis, there will be loss of nuclei. As you can see here, this cell has lost its nucleus, leaving a space, a shadow. كأنما هاي الخلية شبح، لأنه احنا نعرف كل الخلايا لازم بها نيوكليوس، وإذا صارت يعني صار في أي حدث وخلاها تفقد نيوكليوس مالتها راح تكون خلية ميتة. So this is a dead cell. بعد عندنا other features that refer that the cell is dead، ولو هي ما مبينة هنا بهذا السكشن. But we have other features including loss of cytoarchitectural features. اللي هن احنا هنا بالمايوكارديال infarction اللي يهمنا بالcytoarchitectural features under light microscope، اللي يهمنا انه احنا نعرف انه المايوكارديال cells they are striated، يعني بها الخطوط. and these stria من جاية هاي الخطوط؟ جاية من وجود actin and myosin filaments. These stria will disappear after few hours of myocardial infarction because of شنو؟ because of coagulation of proteins. طبعا هذا coagulation of proteins يعني نرجع ونقول أنه coagulation of proteins will lead to coagulation of lysosomal enzymes, and coagulation of lysosomal enzymes will result in inactivation of lysosomal enzymes. That's why the cells are preserved, the cell contour are preserved. بس شنو هي ميتة ومتحنطة. طبعا أي necrosis, أي cell injury will be followed by defense mechanism by the body, by an inflammatory response. And as you can see here in this section, we have dilated blood vessels in the area of necrosis. These are dilated blood vessels, giving rise to neutrophils. Neutrophils are immigrating to the necrotic area, and they will start to invade هذه النيوتروفيلز. هذه neutrophil كل النيوتروفيلز من النقاط السود اللي تشوفوهن. They will start to invade the necrotic area, and neutrophils will start to digest necrotic cells. هذا الموضوع راح نعرفه بالinflammation بعدين بالمحاضرة الجاية. يعني هذا عبارة عن inflammatory response in association with myocardial infarction. This is another photomicrograph of the same case, and as you can see here, there are necrotic cells, remaining necrotic cells. These are remaining necrotic cells, myocardial cells, with dilated blood vessels and